Hi, I'm Ashi from Manhattan High School, and my project is the immobilization of trace elements in wastewater using plant microbial fuel cells. So a little background on microbial fuel cells. They are bioelectrical devices that utilize microbes that are naturally found in soil to produce electrical power. So it is known that MFCs are able to help with the degradation of organic contaminants in wastewater through a series of redox reactions. So these redox reactions should also be able to help with the immobilization of inorganic contaminants in wastewater, such as toxic trace elements, which means that MFCs do have the capability to help with the sequestration of these trace elements in wetlands. Moreover, it can be considered that plant microbial fuel cells do yield a higher potential due to the processes of photosynthesis, creating organic carbon to help with the long-term maintenance of the MFCs, which means that plant microbial fuel cells do have the capacity to maximize the trace element sequestration process. So the questions that I focused on with this experiment would be, would MFCs enhance trace element sequestration? Will the presence of wetland plants enhance trace element sequestration? And finally, will plant MFCs maximize the sequestration process of trace elements? So my hypothesis is that microbial activity induced by the growth of plants will in fact facilitate the sequestration of selenium and lead in wastewater from flue gas desulfurization. So a little background on flue gas. It's basically a pollutant in the air caused by the coal-fired power plants. So basically what they do is the flue gas desulfurization process is treating that air, and then you're left with this wastewater. So my project, the point of this is basically cleaning that water by running it through a restricted volume of soil, and from there it should retain some of those toxic trace elements, so then when you're left with that clean water, you can recycle it for various uses. So the way that I set up this experiment is that I had 12 columns with 4 different treatments, and there was an outlet at the bottom to collect leachate. So the 4 different treatments that I used were soil only, MFC only, soil and plants. The plant specifically I used was actually a wetland sedge, and then MFC plus plant. So the soil that I used was wet soil from a creek bank from the Kansas State North Agronomy Farm. And then how I set up the MFCs, just for your information, is that I had the anode and cathode wires both with a graphite stick, from which I buried the anode wire deep into the soil. The cathode was left on the top, exposed to air to allow there to be more potential between the two. So I watered initially each setup, and then from there I had a regular schedule of watering, a 1-to-1 ratio of FGD wastewater and regular water. And from there I was looking for the electrode potential as well as the concentration of trace elements from the leachate samples. Over here you can kind of see the pictures of my setup and what I did. So over here we have the FGD wastewater and leachate composition. So I tested for 3 different elements, which would be arsenic in the blue, the green is selenium, and then this orange yellow color is lead. So the reason why I tested for arsenic is because the wetland condition does allow for the mobilization of arsenic. So even though there's not much amount, that is the reason why it is being tested for. So in general, we have the FGD wastewater by itself, which is the control, and there's just large amounts of selenium and lead. But through the course of using these different treatments, the best performance is from the MFC plus plant. Even the MFC performs better than soil alone, and then the presence of plants is also enhancing that, and then together they are leaving there to be like, there's leaving an insignificant amount of these trace elements. So up here, if you can see, we have the voltage of microbial fuel cells. And basically with this set of data, I'm just looking for the relationship between the MFCs represented by the blue bar and MFC plus plant represented by the red bar. And as you can see, just the presence of plants does enhance that microbial activity. So some discussion points is that the data over here does not show a constant decrease of these trace elements, simply because I did not recycle that leachate through the system again and again. I added new FGD wastewater. Also, the average concentration of the selenium, lead, and arsenic from the leachate was below the United States Environmental Protection Agency drinking water standard. So again, a lot of that toxic trace element that was retained in the soil. And lastly, the sedges are capable of withstanding low oxygen environments such as the wetlands. So that means that they are producing oxygen through its roots, which means that it is negatively affecting the MFC performance by reducing the gradient of oxygen between the anode and cathode. So which is why there is kind of a decrease in voltage over time, a possible explanation for that at least. So in the conclusions, what I can definitely say is that the presence of microbial fuel cells did enhance the retention of selenium and lead. The presence of plants did enhance the sequestration of those trace elements, and the presence of plant microbial fuel cells did in fact maximize the trace element sequestration process. So plant microbial fuel cells do demonstrate a very promising solution to the cleansing of these trace element rich wastewaters. Thank you.